Hello everyone. Today we are going to see the Biochemistry 8. So in gluconeogenesis, it is basically a reversal of glycolysis, except that there are 4 unique enzymes. And we do want gluconeogenesis especially during the fasting state where there is no intake of glucose, but yet some cells need glucose. What are the 4 key enzymes in gluconeogenesis? First one is the glucose 6-phosphatase. It is the final step in gluconeogenesis where it will be converting the glucose 6-phosphate to glucose, and therefore the name glucose 6-phosphatase. The other one is the fructose 1,6-bisphosphatase. Similarly, this too with the name fructose 1,6-bisphosphatase, it will be converting the fructose 1,6-bisphosphate to fructose 6-phosphate. The other one is the phosphenol pyruvate carboxykinase and pyruvate carboxylase. The phosphenol pyruvate carboxykinase will convert oxaloacetate to phosphenol pyruvate. Pyruvate carboxylase is the one which will convert pyruvate to oxaloacetate, and this is the one which will initiate gluconeogenesis. Let's see that with the help of the diagram. The first one is that which helps in the conversion of pyruvate to oxaloacetate. That is the pyruvate carboxylase. You need to remember that the pyruvate climbs the carboxylase ladder to form the oxaloacetate. Oxaloacetate will form the phosphenol pyruvate by means of phosphenol pyruvate carboxykinase by involving one phosphate in it. Now, these are the first 2 enzymes that are necessary in gluconeogenesis. The other one is the fructose 1,6-bisphosphate has to get converted to fructose 6-phosphate. How does it do? Very simple. Fructose 1,6-bisphosphatase. Similarly, glucose 6-phosphate will get converted to glucose by glucose 6-phosphatase. What are the substrates in gluconeogenesis? Logically, what happens when the patient or when anyone is fasting, then the adipose tissue has to come to our rescue, isn't it? All the fat which is stored in the adipose tissue, the triglycerides which are present in the adipose tissue will act as a substrate. What does the triglyceride contain? The triglyceride will contain the glycerol and the fatty acids. So adipose tissue, in the adipose tissue, it will release glycerol and fatty acids by the breakdown of the triglycerides. The glycerol kinase is the enzyme. So once this glycerol has been released, then the glycerol kinase is the enzyme which acts on the glycerol, converting it to glycerol 3-phosphate. The glycerol 3-phosphate dehydrogenase will oxidize glycerol 3-phosphate to dihydroxyacetone phosphate. This dihydroxyacetone phosphate will enter the gluconeogenesis cycle, which is this reversal of this glycolysis, isn't it? So it will get converted to glyceraldehyde 3-phosphate, which will form fructose 1,6-bisphosphate, and with the help of fructose 1,6-bisphosphatase, again fructose 6-phosphate, glucose 6-phosphate, with the help of glucose 6-phosphatase and glucose. This is how gluconeogenesis is formed with our first most important substrate, that is the glycerol. The next substrate is the lactate. From where is the lactate produced? Lactate is produced by anaerobic glycolysis. So similarly, from this lactate, the cycle can undergo reversal. That is, it can have formed in gluconeogenesis, and the final glucose can be formed. That is by the Pori cycle. The liver is the one which can take up the lactate and convert it into glucose, thereby releasing it into circulation. What is the third most important substrate? Third most important substrate is the amino acids from the muscles. So that is also our primary source during fasting. The amino acids will undergo, after tissue protein hydrolysis, the amino acids will be released, which will get converted to glucose. So what are the main amino acids? The alpha keto acids such as pyruvate, once it is released, it can form gluconeogenesis. But some alpha ketoglutarate, it will enter into the TCA cycle and it will form oxaloacetate, which is a precursor of the phosphenol pyruvate, and therefore gluconeogenesis will be formed. So far we have seen that there are 3 important substrates. So one should not memorize all this. We should have to think whenever a patient is fasting, what is that excess source which is present? First one is the fat, second one is the muscles, and again we have the lactate. So all these 3 are converted to gluconeogenesis by gluconeogenesis into glucose. With our knowledge, let's try to answer these questions. 21-year-old male goes 3 weeks without food, but yet still maintaining stable blood glucose levels. What do you think is the primary source for glucose in this patient? The skeletal muscle protein is the primary source because that will break down into glucogenic amino acids. Liver will convert these amino acids to glucose through gluconeogenesis. Now we know that the skeletal muscle has only releasing the protein amino acids, isn't it? So why can't the skeletal muscle per se convert this gluconeogenic amino acids into glucose? That is because the muscle has a limitation. For gluconeogenesis to happen, we want these 4 enzymes. But the muscle does not have this glucose 6-phosphatase, which is a final enzyme to convert glucose. Therefore, liver remains the primary producer of glucose. Coming on to the another question. 10-month-old boy with growth issues, hypotonia, hypoglycemia, lactic acidosis and hyperalaninemia. So let's go about this with hyperalaninemia. How is alanine formed? Pyruvate, after once pyruvate is formed, it will get converted to alanine and it can also get converted to lactate in anaerobic glycolysis. So since this patient is having hyperalaninemia, that means more of pyruvate is getting converted to alanine. Why? It is because the boy is deficient in this enzyme pyruvate carboxylase. That's what they want to know. What is the enzyme that is deficient in this patient? Pyruvate carboxylase deficiency will cause hyperalaninemia. So therefore, since oxaloacetate and phosphenol pyruvate, and therefore the gluconeogenesis does not happen, this patient is having a hypoglycemia. But why should lactic acidosis be? This is because pyruvate can get converted to alanine as well as lactate. So in pyruvate carboxylase deficiency, there is excess of pyruvate is remaining, which will get converted to both alanine as well as lactate. Therefore, there is hyperalaninemia as well as lactic acidosis because there is deficiency of pyruvate carboxylase. And there is also hypoglycemia because this pyruvate carboxylase is deficient and glucose is not formed. Thank you for listening. All this while, I have an announcement for you. I am starting my mentoring lessons for medical graduates as well as USMLE aspirants. If you like my videos and if you would like to get mentored by me, go ahead, send a message in the comments and fill the Google form. Looking forward to see you succeed. Thank you. Have a good day.